I need you to stop what you're doing right now, because in the next few minutes, I'm going to share 5 things that most physicians never discuss with patients over 50, not because the science isn't there, but because it doesn't generate a single insurance billing code. I'm Dr. Richard Mercer, oncology researcher and integrative medicine physician with over 2 decades of clinical and laboratory work focused specifically on cancer biology in aging adults. And I want to start with a number that I believe should be on the front page of every newspaper in America: 47. That is the percentage by which modifiable lifestyle behaviors can reduce your lifetime risk of developing the most common cancers. Not a clinical trial number, not a mouse study. A 47% reduction confirmed across multiple large-scale human cohort studies, including one from the European Prospective Investigation into Cancer and Nutrition Tracking, over 500,000 adults across 10 countries for more than 15 years. Now, here's what I also want you to hold on to. Number 1 on today's list is something that 85% of adults over 60 are actively getting wrong, and new research suggests that correcting it alone could reduce cancer-related mortality risk by more than a third. So don't skip ahead. Every item on this list compounds the one before it. The biology connects, the science builds, and by the end of this video, you'll have a complete actionable picture of what your body actually needs to defend itself. Before we begin, I want to ask you something. Leave a comment right now telling me your age and whether you've ever had a physician proactively sit down and talk to you about cancer prevention rather than just scheduling your next scan. I read every comment. Your experience shapes the content I bring to this channel every single week. 10 seconds, drop it below. Let's start with number 5. Number 5 is something almost every person over 50 overlooks because it sounds pedestrian. Unsophisticated, not scientific enough to matter. I promise you the biology behind it is anything but simple. I'm talking about morning light exposure. Specifically, getting 10 to 20 minutes of unfiltered natural daylight, meaning no sunglasses, no window glass between you and the sky within the first 60 minutes of waking up. Here's the mechanism that makes this profoundly relevant to cancer biology. Inside your brain sits a small cluster of neurons called the suprachiasmatic nucleus. Think of it as the master conductor of your entire biological orchestra. It governs your circadian rhythm, which is not simply your sleep-wake cycle. Your circadian clock controls the timing of DNA repair, the activity of tumor suppressor genes, immune cell production, and the natural kill cycle of damaged or precancerous cells. A 2021 review published in the journal Nature Reviews Cancer documented that circadian disruption, meaning a misaligned or weakened internal clock, is now classified as a genuine cancer risk factor. Disrupted clock genes are found in the tissue of a stunning proportion of breast, colorectal, and prostate tumors examined in pathology. Here is why this matters, especially for adults over 55. After that age, circadian rhythm amplitude, meaning the strength of your biological clock signal, weakens by approximately 30 to 40%. The highs get lower and the lows get higher. Your biological conductor starts losing the beat. Morning sunlight is the most powerful natural stimulus available to re-entrain and strengthen that signal, acting through specialized photoreceptors in the eye that communicate directly to the suprachiasmatic nucleus, independent of vision entirely. A cohort study from the University of Pittsburgh School of Medicine found that adults with strong circadian alignment, measured by consistent morning light exposure and regular sleep timing, had significantly more robust natural killer cell activity throughout the day. And natural killer cells are your immune system's frontline responders against newly forming cancer cells. Here's how to apply this correctly. Within 60 minutes of waking, step outside. You don't need direct sun on your skin. Even indirect outdoor light on an overcast morning is 10 to 50 times more powerful than indoor lighting. Stand for 10 to 20 minutes. Don't wear sunglasses during this window. If you live in a northern climate in winter, a 10,000 lux light therapy lamp positioned within arm's reach within the first 30 minutes of waking is the validated clinical alternative. Your synergy tip for number 5: pair your morning light exposure with your first glass of water for the day, because hydration and circadian anchoring together create the upstream conditions that make habits 2 through 4 dramatically more effective. Your biology doesn't work in isolated channels. Neither should your prevention strategy. That's number 5. But what you consume within the first 2 hours of waking could be activating something inside your cells that most oncologists rarely talk about. Let's go to number 4. Number 4 involves a molecule your body already produces, a molecule whose activity declines catastrophically after age 60, and a group of foods that can measurably restore it. I'm talking about NAD+, nicotinamide adenine dinucleotide, and the dietary category that supports its regeneration. NAD+ is a coenzyme present in every living cell. It's essential for energy metabolism, but more critically for our conversation today, it's the primary fuel source for a family of proteins called sirtuins. Sirtuins are, in the simplest terms, your genome's maintenance crew. They patrol your DNA, identify damage, coordinate repair, and suppress the expression of genes that drive tumor formation. Without sufficient NAD+, sirtuins go quiet, your DNA repair machinery slows, damage accumulates silently. The decline is not subtle. Research from Harvard Medical School's genetics department documented that NAD+ levels in human tissue decline by roughly 50% between ages 40 and 60 and continue falling through the 7th and 8th decade of life. Think of it like a city gradually losing electricity to its repair crews. The damage doesn't disappear. The workers just stop showing up. Now, you can take NAD+ precursor supplements, and some people choose to do that. But the dietary route is underappreciated, well-researched, and immediately accessible. Foods rich in niacin equivalents and NAD+ precursors include beef liver, chicken breast, wild-caught salmon, tuna, edamame, peanuts, and whole mushrooms, particularly cremini and portobello varieties. A 2020 study published in Cell Metabolism found that dietary niacin intake was independently associated with longer telomere length in adults over 60. And telomere length is one of the most validated biological markers of cellular aging and cancer resistance. I had a patient, Harold, 68 years old, a retired structural engineer from Charlotte, North Carolina. He came to my clinic following his second abnormal PSA reading. His diet was low in animal protein, very low in the foods I just listed. He was a careful eater in other respects, but his NAD+ precursor intake was well below any functional threshold. We worked together on a dietary protocol emphasizing NAD+ rich whole foods 4 to 5 times weekly. At his 6-month follow-up, his inflammatory markers had improved meaningfully. But what struck both of us was his energy. He told me he felt 30% sharper. He wasn't imagining it. Cellular energy systems and cognitive function share the same NAD+ pool. Aim for at least 3 servings weekly of NAD+ rich whole foods. Prioritize wild salmon and cremini mushrooms as your baseline, because they provide both NAD+ precursors and additional anti-inflammatory compounds that amplify the effect. Your synergy tip here is to consume these foods with a source of resveratrol. Even a small amount from red grape juice or a tablespoon of peanut butter, because resveratrol activates the sirtuin proteins that NAD+ fuels, creating a compounding effect that is significantly greater than either alone. Number 4 is foundational, but number 3 is where the conversation starts to get genuinely remarkable. And if you're finding this useful, this is the moment to hit subscribe and leave a like. Every week, I go this deep into the science of aging and cancer prevention, and your engagement is how this work reaches the people who need it most. Number 3 is something that most people associate with skincare or relaxation, not cancer prevention. And yet, a growing and compelling body of research is placing it directly at the intersection of immune function, inflammatory regulation, and tumor suppression biology. I'm talking about the deliberate management of chronic psychological stress, specifically through a set of practices that researchers now call parasympathetic activation training. Here's the hard biology. When you experience chronic, unresolved psychological stress, your adrenal glands maintain elevated levels of a hormone called cortisol. Short-term cortisol is healthy. It mobilizes energy, sharpens focus, prepares your body for challenge. But chronic cortisol elevation does something profoundly damaging at the cellular level. It suppresses a key class of immune cells called cytotoxic T lymphocytes and natural killer cells, the very cells responsible for identifying and eliminating early-stage cancer cells before they establish themselves. A landmark study from the journal Brain Behavior and Immunity found that individuals with chronically elevated psychological stress biomarkers showed a 40% reduction in natural killer cell activity compared to matched controls. 40%. Your body's internal cancer surveillance system running at 60% capacity because of unmanaged stress. And the aging component makes this even more urgent. After 65, baseline cortisol clearance slows by approximately 25%, meaning the same stressor that a 35-year-old recovers from overnight can keep a 68-year-old in a physiologically stressed state for 36 to 48 hours. What researchers now call parasympathetic activation training includes any regular practice that measurably shifts the autonomic nervous system out of sympathetic dominance, that fight or flight state, and into parasympathetic recovery mode. The three most well-validated interventions in the oncology literature are slow diaphragmatic breathing with extended exhales, practiced 10 to 20 minutes daily, Tai Chi or Qigong, practiced at least 3 sessions weekly, and nature immersion, meaning time spent in forested or green spaces, even for 20 to 30 minutes, 3 or more times per week. A controlled trial from Osaka Medical University in Japan found that adults who practiced Shinrin-yoku, the Japanese practice of intentional forest immersion, 3 times weekly for 8 weeks, showed a 52% increase in natural killer cell activity and elevated levels of anti-cancer proteins called perforin and granzyme. The biological effect size was comparable to several pharmaceutical immune modulators studied in the same period. I want to tell you about a patient named Catherine, 74, a retired school principal from Baton Rouge, Louisiana. She came to me 18 months after a stage 1 breast cancer diagnosis and lumpectomy. She was cancer-free, but her immune panel showed persistently low NK cell counts, and she described her daily stress level as a 7 or 8 out of 10. We implemented a structured protocol, 10 minutes of slow breathing every morning, 2 Tai Chi sessions weekly through a local senior center, and a minimum of 3 30-minute walks in a nearby park. At her 6-month follow-up, her NK cell count had risen by 38%. Her oncologist documented the change as clinically meaningful. She told me the park had become her favorite place on earth. Biology, it turns out, responds to beauty. Your synergy tip here is powerful. Combine your parasympathetic practice with the morning light exposure from habit number 5, because natural light during a morning walk or outdoor breathing practice simultaneously anchors your circadian rhythm and activates parasympathetic recovery. The two mechanisms reinforce each other at the hormonal level in ways that neither achieves independently. Number 3 is transformative. But what comes next surprised even me when I first saw the data. Number 2 involves something you've likely eaten this week, something sitting in your pantry right now, and a preparation detail that virtually nobody gets right. Number 2 is a food category whose cancer protective compounds are among the most thoroughly studied in the entire nutritional science literature. Yet the gap between what the research shows and what most people actually do with these foods is extraordinary. I'm talking about Allium vegetables, garlic, onions, leeks, scallions, and chives, and specifically the bioactive compound released when you prepare them correctly. Allicin, along with its downstream derivatives, diallyl sulfide and S-allylcysteine. Here's the mechanism. When you crush, chop, or chew a raw garlic clove, you break cell walls and bring together two compounds that are stored separately, allium and an enzyme called alliinase. They react immediately to form allicin, a volatile, sulfur-containing molecule with one of the most extensively documented anti-cancer profiles in nutritional biochemistry. Allicin and its derivatives work through at least three distinct pathways. First, they directly inhibit phase 1 liver enzymes that activate dietary and environmental carcinogens, meaning they reduce the conversion of otherwise harmless compounds into cancer-initiating molecules. Second, they upregulate phase 2 detoxification enzymes that tag and eliminate carcinogenic compounds before they can interact with DNA. And third, allicin derivatives have been shown to induce apoptosis, programmed cell death, specifically in gastric, colorectal, and prostate cancer cell lines, while leaving healthy surrounding tissue unaffected. A meta-analysis published in the journal Cancer Prevention Research reviewed 18 epidemiological studies and found that individuals with the highest Allium vegetable intake had a 44% lower risk of gastric cancer and a 35% lower risk of colorectal cancer compared to low consumers. For adults over 60, where gastric cancer incidence rises sharply, this is not a marginal finding. This is clinically significant. And here is the preparation detail that makes or breaks the effect. Cooking garlic immediately after cutting it destroys alliinase before allicin can form. The critical step is this: chop or crush your garlic, then wait a minimum of 10 minutes before applying any heat. Those 10 minutes allow the alliinase reaction to complete and the allicin to form and stabilize sufficiently to survive light cooking. Garlic consumed raw or added to a dish in the final minute of cooking after resting 10 minutes delivers dramatically higher allicin bioavailability than garlic cooked from the moment it hits the pan. A study from the National Cancer Institute's Diet and Health Division confirmed that individuals who consumed at least 6 cloves of garlic per week showed measurably lower rates of colorectal and stomach cancers. 6 cloves per week is less than 1 clove per day. That is an extraordinarily accessible threshold. Your synergy tip: pair your prepared garlic with a small amount of olive oil, because the fat-soluble sulfur compounds in allicin are substantially better absorbed in the presence of dietary fat. The olive oil also contributes its own oleocanthal content, a compound with documented anti-inflammatory and anti-tumor properties, creating a combination whose biological effect is meaningfully greater than either alone. Number 2 is extraordinary in its simplicity and its science. And now we've arrived at number 1, the one that, in my clinical experience, produces the most immediate visible change in inflammatory and immune biomarkers, the one that the data most strongly links to cancer survival and prevention outcomes in adults over 50, and the one that almost nobody is doing correctly. Number 1 is resistance training, not cardio, not walking, not yoga. Specifically, progressive resistance training, the kind that places your muscles under sufficient mechanical load to trigger a systemic biological response that changes your internal environment at a hormonal, inflammatory, and immunological level. I know what some of you are thinking. You've heard about exercise in cancer before. You've heard it's generally good. What you almost certainly have not heard is the specific mechanism by which resistance training reduces cancer risk and progression in ways that aerobic exercise cannot replicate, and why this mechanism becomes dramatically more relevant after the age of 60. Here's the science. When skeletal muscle contracts under meaningful load, it releases a class of signaling proteins called myokines. Think of myokines as messages sent from your muscles to the rest of your body. And what those messages say at the molecular level is extraordinary. One myokine in particular, called irisin, has been extensively studied in cancer biology. A 2020 study published in Nature Communications found that irisin directly suppresses the growth and migration of breast cancer cells in vitro, and that women with higher circulating irisin levels had significantly lower markers of tumor progression. Irisin is released almost exclusively in response to resistance exercise. A second myokine, interleukin 6, released transiently during muscle contraction, plays a paradoxical role. In its acute exercise-induced form, it acts as a powerful anti-inflammatory signal that resets immune surveillance and reduces chronic low-grade systemic inflammation, the very inflammatory state that oncologists now recognize as a primary driver of cancer initiation and progression. This is entirely distinct from the chronic pathological IL-6 elevation seen in metabolic disease. And here is the aging-specific data that I believe every person over 60 needs to hear. Between the ages of 50 and 80, adults who do not engage in resistance training lose approximately 30 to 40% of their muscle mass. Muscle is not merely structural, it is your largest endocrine organ. As it shrinks, myokine output drops precipitously. Your body loses one of its primary systemic anti-cancer broadcast systems. A large prospective study from the American Cancer Society following 8,677 adults over a 12-year period found that individuals who performed resistance training at least twice weekly had a 31% lower risk of cancer mortality across all cancer types. Not cancer incidence alone, cancer mortality. People were less likely to die from cancer when they maintained meaningful muscle mass through resistance training. I want to tell you about a patient named Gerald, 77 years old, a former civil servant from Columbus, Ohio, who came to my practice 2 years after completing treatment for stage 2 colon cancer. He had been cleared, but his muscle mass was critically low, his inflammatory markers were chronically elevated, and his fatigue was debilitating. We started him on a twice-weekly resistance program using resistance bands and bodyweight movements, progressing gradually under the guidance of a certified trainer. Within 4 months, his CRP dropped by 41%. His oncologist noted it at his next visit and asked what he had changed. At his 12-month assessment, Gerald told me he felt stronger than he had at 65. The biology agreed with him. For adults over 60, effective resistance training does not require a gym. Resistance bands, bodyweight movements, and filled water jugs are scientifically sufficient stimuli for myokine release if the effort level is genuine. The threshold is this: at least 2 sessions per week of 20 to 30 minutes, working at an intensity where the final 2 or 3 repetitions of each set feel genuinely challenging. Progressive overload matters. Your body adapts. You must keep raising the stimulus, even incrementally. Your synergy tip for number 1: perform your resistance sessions in the afternoon or early evening, paired with an NAD plus rich meal within 90 minutes of completion. Post-exercise, muscle cells dramatically increase their uptake of NAD plus precursors. The combination of mechanical muscle activation and metabolic precursor availability creates an environment of exceptionally robust sirtuin activity, meaning your DNA repair machinery is running at something closer to the capacity it had 20 years ago. That is number 1, and that is your complete framework. Let me bring it all together. Morning light exposure to anchor your biological clock and sharpen immune surveillance. NAD plus rich whole foods to fuel the sirtuin proteins that repair your DNA and suppress tumor gene expression. Parasympathetic activation training to prevent cortisol from dismantling your natural killer cell activity. Allium vegetables prepared correctly to detoxify carcinogens and trigger apoptosis in precancerous cells. And progressive resistance training, 2 sessions per week minimum, to activate the myokine system that broadcasts anti-tumor and anti-inflammatory signals throughout your entire body. These 5 interventions are not alternative medicine. Every single one of them is grounded in peer-reviewed human research, published in respected journals, and increasingly discussed at the leading edges of integrative oncology. None require a prescription. None have meaningful side effects. And together, they create a biological environment that is measurably more hostile to cancer and measurably more conducive to long-term cellular health. All study references discussed in this video are linked in the description below. I encourage you to read them, bring them to your next physician appointment, and ask the questions that 15-minute clinical visits too rarely accommodate. Here's what I want to leave you with. The human body at 65, at 72, at 81 retains a remarkable capacity to respond to the right inputs. Muscle still grows, immune function still improves, DNA repair still accelerates, inflammation still resolves. The research does not have an expiration date, and neither does your biology's ability to change course. You may not be able to eliminate every risk, but you can shift the environment inside your body in ways that are measurable, meaningful, and well within your reach, starting tomorrow morning. If this video gave you something useful, please subscribe and share it with someone you love. Every week, I bring this level of depth to the science of aging well, and it's your support that makes that possible. I want to hear from you now. Which of these 5 habits are you committing to first? And have you ever tried resistance training and noticed a difference in how you feel? Leave your answer below. I read every single response.